Okay, good morning. Welcome back, everyone. It's 10:30, so we're going to get ready with the second part of our morning presentation. My name is Lola Lewis. I am the East Central Urban Forester for the state of Ohio. Please help me introduce our next two speakers. And if I can have our speakers turn on your mic as I get ready to introduce you in just 1 second. Just a little background on their topic, though. We're going to have Dr. Cornelia Leila Pinchot and Dr. Charles Flowers, whose topic today is on restoring American elm to the urban forest. The U.S. Forest Service out of Delaware, Ohio, Forestry Science Lab has been breeding American elm for this resistant Dutch elm disease for over 20 years. Recently, the program Sorry about that. Recently, the program has been expanded to test some of their best American elm genotypes in the urban settings in Midwest and Northeast. Okay, Dr. Pinchot and Dr. Flowers, if you could turn on your cameras, I'd appreciate it, and I'll get you introduced. All right. No problem. Thank you and welcome. First, we have Dr. Leila Pinchot. Current research focuses on the restoration and reintroduction of the iconic tree species, including American chestnut and American elm. Specifically, she studies how to reintroduce tree species to work landscape the content of the larger management objectives. Dr. Pinchot earned her PhD in Natural Resources from the University of Tennessee, her master's in forestry from the Yale School of Forestry and Environmental Studies, and a bachelor's in biology from Oberlin College, right here in Ohio. Next, we have Dr. Charles Flowers, who is a forest ecophysiologist interested in the impact of disturbances on forest ecology system. His current research focuses on understanding elm pathogens, cultivating Dutch elm disease, tolerant American elm, and restoring the iconic American elm across the forest ecosystem. Dr. Flower earned his PhD in biological science from the University of Illinois and Chicago, his master's in ecology, evolution, and organism biology from the Ohio State University, and a BA in biological history from Lake Forest College. At this time, Dr. Pinchot and Dr. Flowers, I'm going to turn it over to you. Take it away.
Great. Thank you, Lola. Wonderful. Well, it's great to be here today. So Charlie and I are going to tell you a little bit about our American Elm Restoration Program, and which, the program in general hasn't had a strong focus on urban forests, but more recently we have a couple of studies that are now we're now getting into that field. So we're really excited in particular to speak to this audience and hopefully get some feedback from you guys. So please ask questions, give us feedback in the Q&A box. We're really excited to have this opportunity to talk to you today. So our program, which, like Lola mentioned, has been ongoing for over 20 years and really based on research that's been going on a lot longer than that, involves a lot of different partnerships, a lot of different collaborators. I can't name them all, but I will name some folks sprinkled through the presentation today. So there are a number of co-authors here you can see on this first slide. I won't name them, but just do be cognizant that a program like this really requires a large partnership of people and entities with different skill sets coming together to try to restore a species. Right. So I am going to start off by taking us through American elm and its ecological and cultural values, talk a little bit about Dutch elm disease, its impacts on the species, and very briefly about historical efforts to restore the species to give context for what we are doing. Then I'll hand it over to Charlie, who will talk about our program to develop CED tolerant populations of American elm. And he's going to tell you about one of our 2 studies looking at elm in urban settings. Then I will come back and tell you about the second study, and then give a few words to conclude the talk, and then hopefully answer some questions. American elm, as you can see in this map, has a very large range, pretty much the eastern half of the United States into southern Canada, found very commonly in riparian areas, although also in upland forests. I've been tromping around the state, different parts of the state. This summer I was down in Tar Hollow in the wilds and saw a lot of American elm and red elm there, and in music, somewhat upland forest. And then I see it very commonly even in the city of Columbus in riparian forest. So even though it's thought of as a floodplain riparian species, it is found in upland forest. And here in Ohio, like I said, it's found in a lot of different ecosystems. So I wanted to get some engagement from you guys, try and make this a little bit interactive. I will go over some of the benefits that the species provides, but before I do that, I want to hear from the audience. So if you'll take a moment to type in the Q&A box what some of the benefits you see that this species holds, either ecological benefits or cultural benefits. If you wouldn't mind typing your response into the Q&A box. I don't believe you'll be able to read other people's responses, but I'll go through and read some of the responses from you just so we can kind of have a dialogue going on. So let's see. Just going to give you a couple of seconds to start responding. Okay. Wow, they're coming in super fast. So not sure I'll be able to cover everything, but thank you folks for being really responsive. Let's see, have canopy cover, habitat, absolutely. And we'll go over habitat in a little more detail momentarily. Very fast grower, yes. Elm is a very fast grower. I can personally attest to that, having watched quite a few of them grow. Practically guaranteed to grow on any site, from Gerald Pink. Wonderful. Yes, we can grow them in our extremely clayey sites here in Delaware. Seen them grow all over the place. When I lived in New Haven, I had them. I grew up right outside of the city, and I had flower pots where I was actually trying to grow American chestnut as part of a study. And the chestnut died and the elm seeded in, and then I ended up just moving the trees when I moved to Tennessee. So brought some elms with me that seeded in. Can handle tough urban situations, exactly. Increased diversity, great canopy tree. Okay, there's way more responses than I can read, so thank you everybody, I really appreciate it. Let me read a couple more. The American elm is beautiful to look at in our neighborhood, and it cleans our air very well. Beautifully said. So with that, I think I'll leave it, but thank you, and I will take some time to go through these later after the presentation. This is really great to have that engagement from you guys. So let me start with some of the ecological benefits, and someone got to habitat, and that's absolutely true. Here, a study from Talamy in Shropshire found that Ulmus, the genus, ranked number 11 in importance for Lepidoptera species. Lepidoptera, of course, are a critical food source for many bird species. There's numerous wildlife species that utilize American elm, porcupine and grouse. Rodents eat the flower buds and the twigs and the bark. Many bird species depend on the seeds that are produced very early in the spring. And of course, as many of you alluded to in your responses to the question, it was a very important street tree, city street tree. because it had that beautiful, or still has that beautiful, graceful form, because it's such a efficient, effective canopy tree, because it can grow in really harsh conditions, because it can clean air pollutants very well. And kind of to sum that up, there's this beautiful quote by Thomas Campanella. Anyone who's interested in the history of American elm in our cities, he has a just gorgeous book, Republic of Shade, a really fun, beautifully written book. He writes, if the American dream had a home, it would be tucked beneath the sunshot canopy of Elm Street, USA. I think that's just so beautifully written. And this map, also taken from his book that he took from a 1934 publication, each dot there represents 10,000 shade trees planted across the United States. You can see just how commonly planted this tree was. Thomas Campanella, in his book, he wrote about how the settlers, the colonists, when they first came to this country, they started planting American elm in their towns and their cities because it reminded them of their European elm back home. And so they were planted commonly in greens in New England and parks, and became places to congregate, places to post signs. And then, of course, they were planted along walkways and streets, and created these beautiful cathedral-like canopies that I think many of us are familiar with. There are poems written about American elm. I'm not going to read this. We don't have time, but a beautiful poem by George Griffith. Paintings that showed the beauty of this tree. And a quick scan of the New York Times archive just shows how commonly it was historically planted, and often to celebrate different occasions or honor Washington's birthday, for example. And the public iTree publication shows that it, like someone mentioned, it's good for cleaning the air, good for reducing particulate matter, good for reducing the temperature underneath the canopy, for reducing carbon monoxide, ozone, sulfur, and nitrogen oxides. So just an all-around really useful city tree. And I was kind of surprised to see this. I was just scanning the literature of for American elm in urban forests, and found that it's still a really common tree, and it shouldn't have surprised me because I've seen it with my own eyes in a lot of the places that I've lived. But for instance, in Grand Rapids, it's still the most abundant city tree there, Grand Rapids, Michigan, 14% American elm, which is problematic because I, in that instance at least, it's not the tolerant cultivars of elm, so susceptible to Dutch elm disease. So getting to Dutch elm disease, this is a vascular wilt disease caused by the pathogens, the fungi Ophiostoma ulmi and the more virulent Ophiostoma novo ulmi. And kind of a neat tidbit of history in the identification of the fungus is these three female scientists from the Netherlands, Dr. Johanna Westerdiep. She's the woman on the left photo, on the right side of the left photo, and she was the first female professor in the Netherlands, incidentally, and the graduate advisor of these two younger women, Dr. Marie Beatrice Scholl-Schwartz and Dr. Christine Buseman. Dr. Scholl-Schwartz was the first to identify Ophiostoma ulmi, so she identified the fungus, and Dr. Christine Buseman, while she was on a fellowship in Cleveland, incidentally, was the first to find it in the United States in 1929. So kind of a neat history there of some early female scientists intimately involved in the identification of this pathogen. And I should mention that it's called Dutch elm disease because it was identified in the Netherlands. However, it probably originated from Asia, so it is not a Dutch species. And I'm going to walk you through the life cycle. I imagine a lot of you have seen this before, but we have a beetle that has the spores of the fungus on its back. It emerges from a dead elm, and we'll get to that point, and feeds in a twig crotch of an American elm. And it is required, an elm needs a wound for the spores to enter the cambium, and this is the wound that creates that opportunity for the spores to get into the cambium. And they get into the xylem specifically, and they will germinate, and they will start to grow and cause cavitation and embolism in the xylem, and the leaves will start to wilt, and eventually the trees will die. The fungus can spread through root grafts, and my colleagues Kathleen Knight and Charlie Flower have a study to explicitly show that, because it's been found through anecdotal occasion, but it hasn't actually explicitly been found through replicated studies. So that's kind of cool that they have that work. So then the trees die, the beetles come back and lay their eggs under the bark of dead or dying elm trees. And the eggs hatch and the larvae emerge, and they burrow their way out, create these galleries, which are really kind of interesting looking. And when they emerge from the tree, they have spores on their body, and then the cycle starts again. So to have elm trees die from Dutch elm disease, you need, obviously, elms, you need the fungus, and you need the bark beetles. And there's native bark beetles, but the European bark beetles are the ones that are a bigger problem. And here's an electron micrograph close-up just showing you the spores of the fungus attaching to the xylem walls, and then the spores germinating in the second photo. And the third photo, this is kind of cool. This is an American elm with some tolerance. I believe this is Valley Forge, some tolerance to Dutch elm disease, and it's producing these tyloses to try to plug up the xylem cells and prevent the spread of the fungus. This is one of the ways that tolerant elms are able to evade the fungus. So fairly early on, there were some pretty intense measures to try to reduce the spread of the fungus, primarily by reducing the spread of the beetle. The USDA enlisted the help of the Civilian Conservation Corps in the 30s to start cutting down millions of elm trees, and they did just that within about 6 years. They cut down, I can't remember how many, but several million elm trees to try to prevent the spread of the beetle, but as we all know, that was unfortunately not successful. A similar approach was tried with American chestnut, and it's just not a successful approach, unfortunately. And my colleague remembers crews coming in and spraying American elm by her in her neighborhood with DDT back in probably the 50s. Which clearly, we now know better. That's not a great thing to do when there's families around, but that was an approach that was used too to control the beetle. And we all know that none of these approaches were effective. And by 1960, over 40 million American elm trees had died. It was probably more intense in urban areas. In rural areas, as I mentioned before, there's still a lot of American elm out there. Many have died, but it's such a prolific species that they'll come back, or some will evade just out of luck, or perhaps also because they have some low level of tolerance. And Thomas Campanella, again, just wanted to read this quote because I think it so beautifully sums up kind of just how painful this was for people living in towns who saw the loss of the species. The towns, once recumbent in the deep shade of trees, now shrank in the blinding sunlight, every crack and pore newly visible in the harsh light. So I'm going to reach out to you guys again. If any of you remember having large American elms in your neighborhood and then remember them dying, please write in the Q&A box, and I will read at least some of the responses, right where the city and state where you observed this. Okay, great. Kate Bradley, Berea, Ohio. Thomas Davis, Cincinnati. Okay, I'm just going to read towns and cities now. Otuma, Iowa, excuse me if I'm mispronouncing that. Jacksonville, Illinois. Buffalo, New York. Large trees in Detroit. die and removed in the 60s. Okay. Northampton, Massachusetts, close to my home. Huntington, West Virginia, and Janesville, Athens, Worcester, Mass, Central New York. Wow, there's a lot of folks who remember elms dying. That's wonderful and very sad. Wonderful just to have this community of folks with that experience, but that length of experience, I should say, but also very sad to have that collective memory. Many more. No, I'm going to stop reading now, but thank you, folks, for who responded. Okay, so very briefly, just want to give you some sense of the really expansive history of trying to restore the species. There were several long-term programs to try to develop or identify American elm that was resistant to Dutch elm disease or tolerant to Dutch elm disease, whichever term you prefer. Starting with Cornell University, they partnered with Boise Thompson Institute and started testing seedlings. They tested over 24,000 seedlings in the, I think, 40 or so years that they were doing this, a really long-lived program. Ultimately, they only found 16 elms that had decent tolerance. Several of those were killed by elm yellows, which is another pathogen which we're not spending much time talking about today, although if you're interested, Charlie is heading a program at our lab to better understand elm yellows and other phytoplasma that are impacting elm. So that program ultimately wasn't largely successful. The University of Wisconsin had a program for a number of years under Ray Geary's, Dr. Raymond Geary's, where they were also breeding American elm, and they developed the multi-clone Liberty elm, which had some tolerance, but I'm not sure if that's available anymore, to be honest. The National Arboretum of Agricultural Research Service, Denny Townsend, pictured here, he actually worked at our lab when ARS was housed at our lab and spearheaded the elm work here for a number of years and then moved to the Maryland, to the National Arboretum, where they developed Valley Forge and New Harmony, two of the cultivars that are widely used today. University of Guelph has a program to find elm trees in Canada, regionally adapted, that have tolerance to Dutch elm disease. And the Morton Arboretum has spearheaded a program to look at other species, primarily Chinese species of elm to use for urban planting. So a long, rich history of work has gone into this with some successes, but it's just such a big problem. And as Jennifer alluded to, we really need, for any tree species that we're restoring, we need just a lot of genetic diversity to make sure that what we put back out, either in the forest and also in cities and towns, is able to tolerate additional stresses. So I think that's where, no, I'm not quite done. Okay. So here's several commercially available American elm that are on the market today, or have been on the market at least. Princeton, and we use that widely at our lab and as a tolerant control. New Harmony, Jefferson, Valley Forge, Colonial Spirit, Prairie Expedition, St. Croix. So there are a number of American elm that are suitable for rural, for city and town plantings. They just don't have the genetic diversity necessary for more rural forest planting. So you couldn't go out and plant, say, 50 Princeton because you just don't have any genetic diversity there. So we aren't there yet. So I'm reaching out to you guys again. If you can tell me if you have planted any American elm cultivars in urban settings, really rural settings either, anywhere. If so, you can raise your virtual hand and or tell us in the Q&A box which cultivars you've used, because we're curious to know. Also, if you face any challenges with planted elms, let us know in the Q&A box, and I'll read through some of the responses. Okay. Let's see. Valley Forge and Parks, awesome. Princeton elms, Valley Forge and Princeton, yeah, those seem to be kind of the two most widely used. Frontier and Pioneer, interesting. I haven't heard a lot about those in Medina. Okay. Numbers can spin. They are very, a very rapid growth indeed. Yes, we have noticed that too. Valley Forge and Frontier. Okay. In Kentucky. Cool. City of Delaware. Where'd that go? Oh, there's Steve Cherry, colleague. Planted Jefferson and Valley Forge recently, also Frontier. Cool. Good to know. Have to see those. I just moved to Delaware. Princeton and Jefferson, New Harmony. Yeah, so Valley Forge. Cool, thank you. I'm going to stop reading so we can continue, but kind of interesting. It seems to be mostly 4 or 5 cultivars that have been used, so clearly not a lot of diversity. We need more, and that's part of what our program will be addressing. So that's the point I wanted to make here.
Well, thanks, Leila, for the handoff, and thanks to everybody for your time and for participating in this. So I'm going to go ahead and describe some of our ongoing efforts to improve Dutch elm disease tolerance in American elm, as well as efforts to restore the species and test methods for reintroduction. So our primary goal here is to produce genetically diverse Dutch elm disease tolerant American elm for reintroduction in forests and urban areas. So one of our sub-goals there is to identify additional Dutch elm disease tolerant American elms for seed orchards and restoration plantings. So these elms for seed orchards need to be produced or need to produce a large proportion of progeny with good disease resistance. We're also interested in identifying Dutch elm disease American elms for horticultural selections. So unlike material for seed orchards, the horticultural selections are sold to people to plant in their yards or for planting along streets. So these are genetically identical clones with good disease resistance, good growth form, and they exhibit other desirable characteristics. We're also interested in testing resistance to Dutch elm disease, resistance of Dutch elm disease to different strains of the DED fungus. And we're developing guidelines for reintroducing elm to rural and urban areas, as well as identifying genes responsible for Dutch elm disease tolerance and many more. So next, we have another audience participation question, so feel free to share your responses in the Q&A box. But we're interested here in how might the needs of American elm for urban forests differ from those of natural forests. And I kind of gave you a teaser on that, but we'll go ahead and let you guys chime in here to see what your thoughts are. And while you do that, I apologize for having to pause my video, but my computer's on the old side and it might glitch up if we let that stay open. So. Yeah, so the responses are coming in here, and so we've got, folks suggest that we have strong wood or branching attachments. We can have construction soils, and they need to be tolerant of some kind of different soils than they would in natural systems. We have less genetic need in urban systems relative to the rural systems, and a lot of other really good responses here. So yeah, as folks suggest, they need to be pollution tolerant, and they need to have good architecture. So moving on, one of our objectives is to identify additional Dutch elm disease tolerant American elms. So my collaborators and our predecessors have established partnerships with elm enthusiasts across the United States. And so this map here on the left kind of denotes the locations of large surviving American elms across the Connecticut River Valley. And 2 of the elms here are shown in the center. And so this material was sourced to us by a collaborator with the Nature Conservancy, Christian Marks. And on the right here is a map depicting the locations of American elms which have been submitted to the Forest Service's Survivor Elm Database, which when we last checked had over 800 submitted trees, which is pretty fantastic. So more information about the Survivor Elm Database can be found here at this website below. You can also easily access it by just Googling Survivor Elm Database. So the website has all the pertinent information for submission, and to keep this manageable, we were asking for healthy American elms that were greater than 22 inches at breast height and in close proximity to dead or dying elms. And so by close proximity, we meant about less than a mile. And so basically the objective of that was to just ensure that these were large residual elms that were on the landscape that had been exposed to Dutch elm disease in the past and subsequently survived. So once we identify a survivor elm, we collect branch material in the spring prior to bud break and produce identical copies back in the lab. So on the left, you can see an image of an arborist collecting branches in New England. So these branches are carefully packaged and shipped to us back in the lab, as you can see here the image of the branches. So we have a residual bank of elm material and have recently sourced 41 survivor elms from New England, Michigan, and Ohio, which were propagated from 2016 to 2017. For our objectives, or for our vegetative and clonal production process, we take the branch material collected in the field and graft this scion onto existing rootstock, as you can see here on the left. So here's a chip bud. And so if this chip bud is successful, or this graft is successful, it grows into new material identical to the parent. And so here you can see our fantastic technician, Nancy, holding this really robust new growth here from a chip bud. So we collect this material, we take cuttings from it and pop those up and baby them in a greenhouse, and then we promote them to a field plantation for further testing. We also utilize controlled pollinations to produce material for progeny tests, in which we cross survivor elms with tolerant elms. We usually try to cross each survivor elm with at least two elm with at least two Dutch elm disease tolerant elms. And our goal here is to identify families that are good general combiners. So our process for these controlled pollinations is to collect pollen from the harvested branches that are sent to us and utilize this to pollinate tolerant trees in our plantations. So as you can see here from the pictures in the top panel, it necessitates gaining access to the canopy, monitoring flowers for receptivity, and bagging flower buds to ensure there is no pollen contamination. So we cut the branches down and with the pollen exclusion bag still intact, and we sort the seeds back in the lab. We plant these seeds, nurture them, and eventually move these to testing plantations or into propagation beds. And finally, after 5 to 10 years of growth, elms are inoculated with Dutch elm disease to test for tolerance. So for our inoculations, we culture Dutch elm disease in the lab, suspend the fungal the fungal spores in water, and inject a small metered dose near the base of the tree. So as you can see here, or as hopefully you've heard, that the time and resources and commitment necessary to produce, grow, and maintain these plantations is pretty considerable. And so that as you can see, this is a slow game to some degree. So following inoculation with Dutch elm disease, we track symptom onset and severity using visual canopy assessments at several different time periods. Traditionally, we use 8-week, 1-week, and 2-year periods post-inoculation. So in the picture on the left, you can see that Dutch elm disease has resulted in significant canopy decline. The image in the center shows a tree with Dutch elm disease and retained necrotic leaf tissue in the mid-canopy, while the tree on the right is exhibiting yellowing and early onset of Dutch elm disease across the entire canopy. And so there's a myriad of symptoms here, but by and large, they result in pretty rapid onset of mortality if the tree is not tolerant. So here we have some results from a lower Midwest survivor elm trial in which clones of 22 survivor elms were planted in complete replicate blocks in Delaware and inoculated with Dutch elm disease approximately 10 years after planting. The figure on the right shows the results of this trial in which we have our elm selections along the y-axis on the left and our 2-year post-inoculation Dutch elm disease-induced canopy decline on the x-axis along the bottom. The bars represent the mean and the whiskers are the standard errors of this. So larger bars indicate less Dutch elm disease tolerance. And as you can see here at the top, the large red bar denotes our susceptible control. The black bars denote our Dutch elm disease tolerant selections, and our gray bars represent the Dutch elm disease, the gray bars represent tolerant by tolerant crosses, and the green bars are the clones from the lower Midwest. So you'll notice some of the survivor trees, for example, SU34 at the bottom and LE21 near the bottom perform as well or better than commercially available selections. We just installed 2 research plantations in partnership with the Columbus Metro Parks at Glacier Ridge and Rocky Fork Metro Park. So here we will assess clones of 26 survivor American elms from the Connecticut River Valley, and we plan on inoculating these trees around 2026. So here's one of our fantastic technicians grinding through, planting out several thousand trees. So this is, quite an endeavor. So we're currently in the process of developing and refining rapid assays to hasten the trial process. By rapid, we mean 1.5 to 3 year assays, which would require far fewer resources, in the form of both time, money, and land. And that's obviously relative to the large plantation assays. So we hope these can be used by partners who want to start their own small testing programs to develop regionally adapted populations of Dutch elm disease tolerant plants. So our breeding progress to date has incorporated over 40 large survivor elms from New England, over 30 from the lower Midwest, and 4 from the upper Midwest into our breeding program. We've installed 3 test plantings that will be inoculated between 2020 and 2025. And as we mentioned, we're continuing to refine these rapid assays and continuing to add new survivor elms into the mix. So moving on to some of our reintroduction efforts, we've been using selected American elms in a variety of restoration efforts. So we've used these in emerald ash borer impacted floodplains to formerly grazed riparian corridors and reclaimed mine lands. So you can see here on the bottom left, these are 2 images from the Finger Lakes National Forest in upstate New York. And so you can see traditionally they have these cattle pastures where cattle are allowed to just graze all the way down to the riparian stream itself. And so we're reinforcing these systems with elm as well as other species to reestablish some ecosystem properties here. And we're also, Kathleen Knight has been utilizing elm as well as some other species in EAB impacted floodplains. And here you can see an American elm on the right standing in a seasonally flooded pool of water at the base of an ash tree. So it's great to follow up Dr. Cook and Knight's EAB ash breeding talk, so I don't have to dive into great detail on EAB. But in case you missed the talk, the emerald ash borer is an invasive tree boring beetle which has decimated ash trees across the United States. A recent modeling simulation by Iverson et al. Identifies American elm as a species well positioned to benefit from EAB-induced ash decline and climate change. Furthermore, some parallel research in the bar graph on the left indicates that elms do indeed co-occur with ash in forests near the EAB epicenter, and they exhibit rapid radial growth in conjunction with ash decline, indicating that indeed this species does respond and is present in these systems. So additional research by Looney et al. From EAB decline simulated wetlands in Minnesota suggests that American elm survives very well in these systems as well. So all in all, elm is historically a really important species across these riparian systems, and it's after it declined, ash kind of moved into these systems. So it's a logical species to get back into these. So in contrast to the rural reintroductions, urban reintroductions have unique challenges, as we've previously discussed, and we'll reiterate here. So we've got harsh environmental conditions denoted by heavy compaction, contaminated soils, heat island effects, construction, vandalism, competition from invasive species. We also require some unique aesthetic and structural requirements for urban trees, and we require higher levels of disease tolerance as well as higher levels of survival. So there's some slightly different characteristics of these urban trees relative to the rural. In 2018, we initiated an urban riparian forest restoration project at 5 sites across Columbus, Ohio. As you can see from the image on the left, we have a dense understory of honeysuckle, which is common in these riparian systems in Ohio. So it can form dense layers resulting in dramatic changes to the ecosystem. It does so through direct suppression of native plants, altered nutrient cycling, and shifted structural complexity. And so you can see here are 5 sites across central Ohio. So we have sites at Casto, Tuttle Park, Kenny, Three Creeks, and Wolf Park. So our overarching research goal for this project is to control invasive shrubs, enhance biodiversity, and habitat quality. We're interested in enhancing ecosystem function via the removal of invasive species. We're interested in investigating the relationship between vegetation structure and complexity, as well as avian abundance and behavior. We're interested in reintroducing native woody plant material, specifically elm, bur oak, pawpaw, musclewood, coralberry, and nannyberry. And we're determining strategies and guidelines for riparian restoration. So at each of these sites, we delineate 4 plots. One of the plots receives a invasive species removal and planting treatment. One gets no treatment, it's a control. One gets planted with no invasive removal, and one is an invasive removal with no planting. And this project's in collaboration with Steve Matthews and Alex Fotis, as well as the City of Columbus and the Columbus Recreation and Parks Department. So I'd like to use this slide and the next one as a quick shout out to our partners at the City of Columbus and Columbus Metro Parks, who not only made the research sites available, but also assisted with the invasive clearing and the herbicide treatment. So you can see here, a diligent city employee chainsawing down some dense honeysuckle, and you can see here on the right, prior to our manipulation, along a heavily utilized bike path in Columbus, just north of OSU's campus. But you can see off to the right and the left, dense understories of honeysuckle, which clearly are in dire need of some attention. So again, we've got some post-honeysuckle removal pictures here. So you can see on the left the cleared understory, and in the center here and on the right, planted individuals. So it's a bit early in the experiment yet for any conclusive results, but we're tracking the survival of planted restoration material. We're tracking vegetative growth, successional dynamics, changes in canopy structure, and we're using a handheld understory LiDAR for those measurements, as well as tracking wildlife impacts. So here's some early results from this, but the figure on the left shows that 1 year following planting, on average, there was higher survival of the planted seedlings in the invasive removal plots versus the control plots. So that that's not surprising. The understory species adapted to lower light conditions and exhibited some of the best survival. And so you can see here that our objective was to basically try to utilize some techniques to minimize that reinvasion of honeysuckle. And so one of the tools that we used there was planting overstory species. So we used bur oak and elm. We have a mid-canopy species that we use, 2 of them, musclewood and pawpaw, and some understory species, nannyberry and spicebush. And so we've got pretty good treatment effects there, and marginal differential between the different species. Within the elm itself, we've got some differential responses of the elm. So we've got the 6 selections of elm that we've utilized in this study, and we have some differential selection. But by and large, most of them perform a little bit better in the removal locations versus the non-removal locations. Next up, we have some preliminary figures that highlight the shifts in forest structural complexity associated with honeysuckle removal. So on the far left, we see the average vertical distribution of vegetation, and so that's across these 2 different sites. So the far left figures, the gray lines in those figures denote the pre-removal distribution of vegetation, while the red lines reveal the post-removal patterns. You can clearly see the reduction of leaf area at about 7 meters associated with the honeysuckle removal. These 2 figures stacked on top of each other in the middle represent basically the LiDAR transects or HIC grids, where red boxes denote the loss of vegetation. So the removal transitioned forests from bottom heavy to more structurally even systems. The figure on the right is a principal component analysis where the axes describe relationships between environmental space described by canopy structure, and the circles represent 95% confidence ellipses. So I don't want to belabor this this figure or get into the weeds on it too much, but basically you can see that the the honeysuckle removal created a situation where we have increasing vertical evenness and more diffuse leaf distributions, as well as higher structural complexity. And so these patterns have been linked to increased resilience and increased carbon storage. And one of the things we did for this study, we layered in adjacent plots that Amanda Rodewald and Steve Matthews utilized for a bird study. And so they conducted some removals 10 years ago. And by and large, the trajectory holds up for our control plots, our 1-year removal, and the 10-year removal plots. So, which is really, really helpful to see those clear patterns.
Great. Okay.
So I'm going to pass this back off to Leila.
Well, I want to introduce our newest study, and the second study that we've established to look at planting elms in urban settings. The study Charlie just mentioned, obviously that's in urban forests that are a little more wild in terms of the vegetation is left to do its own thing. This study that I'll be describing is we planted American elm in park settings, so managed settings, more kind of urban-like settings that you'd typically think of. And this is in collaboration with Vince D'Amico. He's also Forest Service, and Tara Trammell and Danielle Ecologist, both at the University of Delaware State. So basically the point of this study is we're interested in looking at how some of our best performers in terms of DED tolerance also perform in urban settings where they have those additional stressors like compaction and heat and higher percent impervious surface and pollution, etc. We want to see if some of our really highly DED tolerant elms can also do well in those sites over the long term. And so here's the same graph that Charlie showed you a few minutes ago. So I'm not going to go over it again, but basically, we're looking at percent canopy decline. So the bigger the bar, the worse the tree did. The red is the susceptible control, and the black, Princeton and Valley Forge, are the tolerant controls. We chose these three selections that tested really well. Obviously, they're comparable to Valley Forge and Princeton in terms of tolerance. And they propagate really well, which is why we chose them, because they're good propagators. Some of our trees that are highly tolerant don't propagate very well, and obviously that won't make a good cultivar if you can't create more trees. And we work with those partners in Newark and their partners in Philadelphia. The Philly Field Station is a Forest Service research station based in the city, and they collaborate a lot with the University of Pennsylvania. So we had a really good network of folks who are interested in getting elm back out into an urban landscape, and had partnerships with municipalities and nonprofits and other collaborators locally. So it's just this great network of collaborators where we could find a bunch of different sites to plant these selections. And what we wanted to do was within each city, and really across the 3 cities, but specifically within each city, is create this gradient of urbanness, if you will. And we specifically use percent impervious surface within 50 meters of a potential study site to calculate that. So we wanted some sites, all city parks, so they're all going to be quote unquote urban, but we wanted within that there to be a gradient of percent impervious surface. So what we did is we worked with our partners, and within each of these cities we selected, or we identified a number of sites. Here in Columbus, we identified nearly 50 potential planting sites. This slide, obviously you can't read it really well, and that's fine. The point here, this is percent impervious surface on the x-axis, and the point just is to show you that gradient. Each of these dots represents one potential study site. Now, the available study sites ranged from close to 0% impervious surface to about 80. And what we finally went with, and this is just 10 of the sites, I apologize, I don't have the data available for the 5 immediately, but they range from about 4 to 60% impervious surface, the sites here in Columbus. The sites in Newark and Philly differ a little bit, but I'm not going to show those data. And we worked very closely with the city of Columbus. They were just wonderful to work with. We spent a lot of time identifying potential sites for them. Rosalie Hendon, Tina Moan, and Craig Seeds were really, really wonderful to work with. Columbus Metro Parks, Andrew Booth and DJ White, just wanted to give a shout out to these folks. Green Lawn Cemetery, Randy Rogers, that's right, in the heart of the city. City of Powell, just north of the city, but in the greater Columbus area, by the Bowers in the city of Dublin, Brian Goodall, were really helpful in helping us identify potential sites. And the 15 sites that we eventually did select because they represented that range, gradient, and because they also just represented a geographic range were these 15, for those of you who are familiar with Columbus and the parks here. So you can see they're kind of all over the place, which is exactly what we wanted. And I don't actually have any data to share with you because this was planted so recently, but I just wanted to introduce you guys to the study because it's new and because we hope to expand it, and we're always looking for new ideas. So I wanted to get you guys introduced to this. So here's the study site with the lowest impervious surface in this park. You can see on the left the map, there's that cluster of yellow dots. Those are the planting sites. And the closest impervious surface here is actually a walking trail. So, as rural plantings go, or as urban plantings go, this is pretty rural. It's right on the forest edge. And it'll be interesting to see how the trees do there versus a site like Scioto Audubon Metro Park. While this looks like there's a lot of grass, immediately in front of those trees is a big old parking lot, which is attached to the one of the roads in the Metro Park. This is the cluster of yellow pins right by the entrance of Scioto Audubon. And for those of you who are familiar with this park, these trees were planted right by the main office. It's not the nature center, but it's actually the main office where a lot of the staff is located. So it'll be interesting to see how they do over time. So we planted these just under a year ago, just 3 selections. We kept it pretty small. This is kind of a pilot study to get our methods down and build these collaborations. And in each of the 3 cities, we just planted 45 trees, so 3 trees at each of the 15 sites. So fairly small. We do a lot of rural restoration plantings where we might have 1,500 trees in a study. So this is a much smaller, different type of study. But obviously each tree we're babying a lot more than we would for a rural restoration study. We have these cages with T-posts and tree diapers that we eventually mulched. We watered them a couple of times this year because we had a really dry early summer, and the trees were showing signs of stress. So it took a little bit more work than some of our normal restoration studies, but we're really excited to see the data over time. So just out of curiosity, raise your hand. There's a little person with a raised hand above the presentation you can see. Raise your hand if you've used these tree diapers, or if you prefer, write it in the Q&A box. I'm just curious to see how many folks have used these. Okay, at least one person I can see in the Q&A. I actually don't know how to see who has raised their hand, so maybe Tyler or someone else can provide that information later. Cool, so there's a number of people who have used them. Some people use tree gators, similar concept. Yeah, so these were new to us, actually suggested, I think by Rosalie, and I'm really pleased with them. It has been a really dry year, but we've only had to water them twice, and they've retained a lot of moisture. Cool, thanks folks. I'll read through those a little slower. after the talk. But yeah, so we've had good luck with those. Like I said, we mulched them in just to retain the moisture a little bit better. After these pictures were taken, we did that. And they seem to be working pretty well because we just don't have time to water the trees very frequently. So we will be looking at simple things like survival and growth. Also, we will be at each site within the next week or two. We'll be putting out ozone samplers to get an idea of the ozone concentration across these sites. And then we're looking at stress signals within each of the trees. So we're looking at metabolite levels that might indicate stress, looking at chlorophyll levels, also isotopic carbon and nitrogen, which can indicate drought stress and other stressors, just to see if stress indicators correlate well with percent ozone or percent impervious surface, and just get an idea of how these trees are doing in different sites. I mean, we all know that American elm does well in cities. Obviously, there's a long history in our country to prove that, but there could be variation in individual elm. And for these urban plantings, we really need trees that have high DED tolerance and high tolerance to these urban stressors, especially if they are impacted by DED in the future. If they can't tolerate urban stressors, both of those stressors together could reduce their ability to tolerate either stressor. So we're looking really for the best of the best, and that's why we're testing these. So only 3 selections at first, but like I said, we are starting to talk about expanding the study. We have received some funding to do so. We're not sure if that means looking at additional sites, but certainly additional genotypes. So if this is of interest to you, if you have sites, really we're probably not going to leave central Columbus or central Ohio just due to logistics. But if you're in this area and have an interest in this type of project, please reach out to us. I don't think I provided my email, but leave a message in the Q&A, or you can Google either Charlie or me pretty easily and find our contact information. Just a few pictures to show you. There is a lot of variation already in how the trees are responding. I think the drought stress took a toll on some of them. We've had a couple die, so we will be replacing those. I think transplant shock doesn't really count as we need them to at least establish before we can really look at their response to these conditions. But some really nice healthy trees on the left, and then a tree with some dieback on the right that doesn't look so happy. In one of the sites, that's Toyota Audubon, that has the higher impervious surface, and the trees there do look like they're suffering. So I just wanted to take a couple seconds here to take a big step back and comment on our program. We've talked, we spent a little bit of time talking about the program, but I wanted to just think about it holistically. So ultimately, we want to develop these populations, genetically diverse American elm, that can be used both for rural and urban plantings. Here, this map shows all of the different plantings that we have, and also some of the dots are survivor elms, so it's a little bit deceiving. But I know it's probably hard for you folks to see these dots, so I'll stick with the big ones, the big orange dots that are in central Ohio and New England. Those are our resistance plantations. So those are our field plantings where we plant survivor elms and then inoculate them. So we have 4 of those in the Midwest and New England at this point in time. As Charlie mentioned, it was a really, really labor and time intensive to keep those going, and it requires keeping them in the ground for over a decade before we can assess the tolerance of those trees. So a lot of work, but that's kind of the backbone of our program. That's where we identify the trees that can then be used in rural restoration, and now more recently, like we talked about today, some of our urban restoration trials. Yeah, so that's kind of where we are now. We're always hoping to expand, to test new survivor elms. Moving forward, we're hoping to develop a model where we can work with partners to establish some of their own resistance plantations, and test some of their own survivor elms, because as Charlie kind of mentioned, we're just about at capacity. It's a lot of work, and we pretty much have our hands full. So we're still thinking about how to do that effectively, but that's kind of the model that we're thinking about moving forward. Also, we're really focused on the Midwest and New England, but obviously elm had a much bigger range, so we need to think about how to move into other areas working with partners. And with that, just like to acknowledge folks, a lot of our funding comes from the Manton Foundation, which is based in Boston, and they've just been such a tremendous asset to us. We can't thank them enough. Del Lessor is a really wonderful farmer in Michigan who's been working with the program at least 15 years and bringing survivor elms to us, which has just been tremendous. And the Nature Conservancy, also just a great partner, and I mentioned some of these others. So thanks to all of them, and thanks to you for your participation today. And with that, that's all we have.
All right. Thank you, Dr. Pinchot and Dr. Flowers. Before we get to the Q&A, I just want to remind you all to join us tomorrow afternoon and keep your eye out for today's evaluation in your box. It's very important that you respond to the evaluation. That helps us do this type of program for you and find out what we did, what we need to do, and what you're looking for. So we'd appreciate it if you would fill out those evaluations. If you don't receive it by the end of the workday, please contact your regional urban forester and they will email it to you. Your feedback, again, is really important to us, and we appreciate it tremendously. We will also have a special post-conference, the Coffee Clutch, on September 30th for anyone who wants to join in a casual conversation about what you learned or what you would like to know more about. The link for that is at the end of each weekly evaluation that is emailed to you. Now, at this time, if I can get Kathleen and to come back on, if she's still here, to have Dr. Pinchot and Dr. Flowers to come back in to do the Q&A part. Okay, thank you. One of our questions was from Jason, and he said, seeing some new elms growing too fast for their own good within a tight angle of attachment. Do you know of any cultivars that are doing this?
Really delved into looking at branch architecture. I know that the folks at University of Minnesota, that's kind of their forte. So I can't comment on that unless Charlie or Kathleen, who's a key collaborator in this work. And
yeah. That's kind of outside of our realm, but yeah, I agree with Leila that the like Chad Gibland and those folks up in Minnesota have done some work on that and found, I think they saw a lot of tree damage in Valley Forge.
Kathleen, did you have something you want to add?
But yeah, that would be a better question for them.
Okay. Well, thank you for your response and stuff, and we'll, again, try to maybe get a link to the folks up there.
Yeah, it's here. I mean, we have so much native elm, not just American, but other species of native elm that host the fungus. So it's here,
trying to eradicate the cobalt.
It's here to stay. Unfortunately, there's no way to eradicate it. Yeah.
Thank you for your response. Does anyone else have
Yeah,
any input
typically
on
once
that?
fungal pathogens are in the landscape, they're pretty well
Consensus across the
established.
board? All right, we'll move on to the next.
So with ash, we've
Thanks, Charlie, for your
actually
input.
we've collaborated
The
a
next
little
question
with
is,
some
how does
folks who
current
have
knowledge
expertise on that.
that
We don't
trees have
have
internal
kind of final
residential
results on that
fungus
yet.
affect your research? That's a good question.
Yeah, and with elm, we're really focused just on the response to the pathogen in very, I shouldn't say very controlled settings because they are field plantations, so they're not controlled. But yeah, we haven't looked at other other organisms and how they might interact with the fungus. I think that would be interesting. We're so focused on just trying to nail this because there's still a lot to be answered about just the fungus itself and how to appropriately rate its impact on a tree. But I think that's a really useful point, something to look into.
Well, thank you for that. Does
There
anyone
are a
else have
number
anything you
of
want to add?
commercial nurseries
All right.
that
We'll move
do
on
sell
to our
those
next one.
those
And the next
collections that we
question
mentioned,
is,
those
where
cultivars
can
that
I
we
obtain
mentioned
the
in the
DED
presentation.
tolerant
I
American elm to
I
plant
can't
and
name
grow
them
in an
offhand.
urban area?
I'd have to look in my notes to see, because we've tried to obtain them, and I can tell you it's actually difficult when we've tried to purchase them for to include in experiments. We've been told, I think like Schmidt and Sons was one of them, that there is a 1 or 2 year wait list for trees, so they're in high demand. So offhand, I can't name the nurseries, but there are a number of nurseries that sell these different cultivars.
All right, and thank you for your response. Appreciate it. Another question is from Matt.
I personally haven't.
Valley
Charlie or
Forge.
Kathleen, have you?
And what he's saying is that
No, we've
have
been lucky to
lost a
not
few
have wind
trees and many
damage
larger
in
than
some of the
wind
places that we've
damage.
planted.
Have you seen it in any of your
Our
other
trees may
research
just
areas?
not be big enough. But
Yeah,
as
most of our
Kathleen mentioned
plantations
before,
are
the
pretty well
folks at
sheltered too,
University of
and
Minnesota,
so we don't,
that's
and the trees
precisely
aren't
what
quite as
they
large, and so we
found
haven't
with
experienced much wind shear
trees
or anything.
planted throughout Minneapolis, St. Paul, is that there was a lot of wind damage to Valley Forge.
Okay. All right, I have a question for you and stuff, and again, and it's specifically to the respective research that you've been doing, whether it was with the ash or with the elm tree and stuff. Have you found any areas that have been
Do you all
really
want to
challenging
tackle that
for
first
you?
for elm, or do you
And
want
if you can
me to
identify
jump
those
in for
areas
ash?
and tell me some of the things that you've done to overcome some of the
If
challenges
you
that you've
have
faced
something
in the process
in
of doing your
mind, go
research.
for it.
Sure. So for ash, one of the big questions that's a challenge for us right now is figuring out how to
Kathleen, would you like
select
to start?
surviving ash trees that are worthwhile for Jennifer to test. So, that testing process is so labor intensive and so expensive that we really want to try to be testing trees that have a good likelihood of actually having resistance, and not just testing trees that are kind of the last to die in a site. So one of the challenges that I'm kind of working with doing data analysis, pulling in data from a bunch of my monitoring plots in Ohio, monitoring plots in Pennsylvania, and then taking a look at some of the FIA data that's available elsewhere, is just trying to figure out, what size threshold should we be setting for it to be considered a surviving large tree for lingering ash, and how long after emerald ash borer has gone through, and for places we don't have data, how can we tell how long ago that was, so that we can be sure to be selecting trees that have that good likelihood of success. And we'll do some dendrochronology studies as well, I think, in Michigan over the next few years to really get a good handle on that, so that we can tell people, when they call and say, I have a surviving ash, we'll have even better criteria than we have now for saying that's a tree of interest that we would really like to test.
Okay, well, thank you. Before I go on to the next question from our participants.
Yeah, so we've been working through different, complicating factors as far as elm goes. We ran into a phytoplasma issue that we've been trying to get to the bottom of. That's kind of overlaid in the background of our DED kind of scenario, and so that's one of the things that we've been working with. But we're kind of trying to look at, one of the biggest things that we've been struggling with is this rapid assay and trying to look at kind of unique or novel tools to identify Dutch elm disease tolerance early on. And so, the kind of overhead associated with the infrastructure for the plantation tests is just too costly in the long run. And so if we could streamline that process, I think that would enable us to push more material through the testing system and elevate useful candidates earlier in the process. So hopefully we can really enhance these efforts moving forward.
Really nicely.
All right, I appreciate that you sharing that. Leila, did you have anything you want to add, or are you good? All right. Hey, Kathleen, I have a question for you from Tom. He says, I frequently dig up ash and elm saplings and transfer them.
We definitely have shown that deer like to eat elms. So in all of our experimental plantings, deer protection has shown to be very helpful for
have you had any of your sites
restoration plantings, and then any of our experimental trees in some of these larger experiments, we fence them in with big fences to make sure that we can do the experiment without the trees all getting eaten. I haven't tried to transplant any elms, but yeah, the deer are an issue. So the larger ash trees get killed off by the EAB, and there are smaller ash seedlings and saplings that are left behind that are either a less preferred smaller size class, or they're just too small to get infested. But as they grow, emerald ash borer can infest those over time, so it's kind of surviving on that last little cohort of ash as they get larger. It can also, infest these surviving lingering ash trees. They don't kill every ash borer that attacks them, so they can, occasionally attack those as well. So they're surviving at really low levels on that remaining ash that's there.
Okay. Thank you, Kathleen.
I'm not sure if Frank is
The American
talking
chestnut
about
is a cultivar.
pure
I have
there's
seen
plenty of places
some
that sell
online,
pure
but not
American
sure
chestnut that
if
are
they are
not
legitimate. So,
resistant to blight.
have you run across any false
And
American
they usually
chestnuts, or
will,
can you make a
will say that.
recommendation?
But I've planted them on our family property just because they usually produce nuts before they get blight and die, or sometimes I should say. I have seen, for instance, the Dunstan chestnut is really mostly Chinese chestnut, and it's been marketed more as a hybrid. But so that's the only example I can think of where it, maybe isn't quite factual. But there are no, highly American hybrids available commercially, because there really are no highly American hybrids in existence that are, that are resistant to blight. That program is still ongoing, and it suffered some setbacks, but they're going to get there. So in the meantime, I suggest planting either pure American, knowing that eventually they'll succumb, or I've planted pure Chinese on our property too, in food plots, because we have a hunt club. But that's, I guess, up to your personal preference. So,
well, thank you for your response.
I guess the easy answer is convention. That's just how our lab has done it for years. But there is the thought that there could be juvenile resistance. So younger elm trees may just inherently be more resistant to or tolerant to Dutch elm disease than more mature elm trees, and we would we have to get past that phase before we can test their true tolerance that they will exhibit when they're mature and planted out in cities or in more rural settings.
Okay, thank you. Laura,
Sorry, can you
her
repeat the
question is, what is the approximate success of the American elm sold at commercial nurseries in the urban area? Do any of you have the answers on that, either Charlie or Leila? Is that something y'all covered?
Oh, I'm not sure that anyone, tracks that. So I'm not quite sure how to answer that.
What is the approximate success of the American elm
Charlie or
sold
Kathleen, do you
at
have
commercial nurseries in the urban area?
I mean, I think if there are these tolerant selections, or these, these ones that have been pretty thoroughly tested, the tolerance mechanisms are reasonably robust and they should hold up pretty well. But, if they're not these cultivars or these selections, they're probably reasonably untested.
Okay, there is in a publication from a few years ago, we have an American Elm Proceedings publication, and in that there's a list of available cultivars and any information that's been published about their respective tolerance. So that doesn't quite get to the question, but if you're looking for information about the tolerance level of a specific cultivar, which, as Charlie suggested, should give you an idea of how they'll do, at least in the face of Dutch elm disease, that's a good resource. So American Elm Conference Proceedings has a nice cultivar list by Linda Haugen and Susan Bentz from the ARS.
And I will point out, Steph Miller responded there in the Q&A and said, consider talking to your regional urban forester. We often have a feel for how trees are performing as towns use them. So that's another wonderful resource.
Thank you, Kathleen. All right, a couple more questions, and then we'll kind of wrap it up.
No, I am
Here's
not.
one from Rita McKenzie. Have you tried
Kind of
using BioBarrier as a moist retainer?
Like,
Moisture retainer.
do they mean like using it on top of the ground, kind of like landscape fabric?
She goes further to state that we have a project at Purdue. I'm sorry, go ahead.
Okay.
No.
Yeah, I haven't used that.
I would assume as much. It's just how she stated, have you tried using a BioBarrier as a moist retainer? But she also further said,
No.
we have a project at Purdue looking at what type of mulch, yes, assists in longevity. The trees
Nice.
with
Yeah,
the
that
barrier
sounds like
loam
something to
do
look
better.
into.
Go ahead, Leila. Did you have something you wanted to input? You have not. Is your response? Okay. All right. Yeah.
Well, so they're pretty different. The American chestnut breeding, which is currently spearheaded by the American Chestnut Foundation, they're breeding Chinese chestnuts with Americans. So they're using resistance from the Chinese chestnut and trying to breed that, with the American, and then back cross it with several generations of American to kind of breed out the Chinese characteristics. And we actually aren't doing a lot of actual breeding, like crossing the trees together yet. Charlie did mention some of the progeny tests that we have, which function really just to test the survivor parents, not actually to breed. And also, we won't be hybridizing. So like I said, Chinese or chestnut, they're hybridizing two different species. With American elm, we're just going to stick with American elm. So it'll be more similar to at least some of the work that Jennifer mentioned of just breeding together like the best of the best elms. So we're not hybridizing the species. So pretty different in that respect. But same in that it's a really long process with a lot of stumbling blocks. Well, it's harder to hybridize American elm with the Chinese elm. I think the biggest issue, if I remember correctly, is that they
he also wants to know,
they may not be compatible, but also they flower at different times of the year. So that really isn't an option for elm. And we are having better success based on our program of finding some pure American elms that seem to have high tolerance, at least based on our testing. There's been some American chestnuts that have been found with okay tolerance, but I think the audience member who asked that is spot on, that we will have better success with American elm. At least that's what we're hoping. Yeah.
Okay. All righty. See. Theodore wants to know, what is the metabolic level?
So some of the metabolites that we've been using in this, we're kind of in the preliminary stages of that, are polyamines. And so specifically, we've been using putrescine, whose cellular concentrations kind of fluctuate over baseline levels based on background stress rates. So we're we run a whole suite of metabolites, and we're kind of cherry picking the best ones to provide us the signal. So.
Okay. Right, and I'm going to end, which I think you've probably answered this.
So it can infest white fringe tree as well, but that's the only one that they really know of other than all the species of Fraxinus.
Any alternative hosts for the EAV? All right. Well, I just want to thank our panel for their response to the questions. Everyone has done a great job today. For the session from the USDA Forest Service, Dr. Knight, Dr. Koch, Dr. Pinchot, and Dr. Flowers. And I want to thank you all again that's out there, our participants that joined us today, and we are looking forward to seeing you tomorrow as well as next week. So thank you.
Thanks, everyone.
And for those of you, again, just to reiterate, as far as the ISACEUs, they
Thank you.
be given at the end of the series. Thank you.
Thank you.
All right, bye. Thanks, Kathleen.
Thanks,
Thanks,
guys.
Leila. Thanks, Charlie. Fantastic.